So this is a kidney like we were just looking at, so that we're seeing the cortex and we're seeing the renal pyramids, the medulla, we're seeing the renal columns, and we're seeing the collection system, the minor calyx, major calyx, renal pelvis. And so one of the reasons why the cortex and these pyramids look strikingly different is they contain different parts of the functional unit of the kidney, which is called a nephron. So if we look up here, we can see we've got these little white balls that we see that are only in the cortex. We don't see any of those down here in the medulla. So this is called a Bowman's capsule, and it's the place where filtration is going to occur of blood to make urine. And then from this we have a duct system that's going to help process the urine. And so as we get into the pyramid, we can see these white ducts that are coursing down through the pyramid, coming back up, and then coming back down the pyramid again. And so these are all ducts associated with a nephron. So the pyramid is made up of millions of these ducts aggregated together, where the cortex is actually made up of millions of these round structures that we call Bowman's capsules, that are where the filtration occurs. So if we take this part of the kidney with the associated cortex and pyramid, then we can see in more detail what we're actually looking at here. And so each of these round things is a Bowman's capsule. If we cut them open, then we have a capillary bed in here where the filtrate is produced, which is called a glomerulus. And then associated with the Bowman's capsule is a tube that's highly folded. And so since the tube is folded, we say it is convoluted. And we're going to use words that we used on appendages, proximal and distal, as a relationship to the round thing. So since this tube is actually connected to this Bowman's capsule, then it becomes a proximal convoluted tubule. Now, convoluted tubules lead to these long linear tubes that drop all the way down through the pyramid and then round and go back up through the pyramid. So this entire loop that we're following here is called a loop of Henle. And then what we do is we follow urine filtrate flow. And so from the proximal convoluted tubule that we talked about up here, the urine is going to flow down through this tube. So this is said to be the descending limb of Henle. And then it's actually going to curve at the bottom, which is the loop of Henle down here. Then the filtrate is going to pass back upward. So over here, this is said to be the ascending limb of Henle. Now notice that they made this look thin, and this look thick. So what we're actually going to do is we're going to call this the thin descending limb of Henle. We're going to call this the thin ascending limb of Henle. And then here where it looks thicker, we're going to call that the thick ascending limb of Henle. And in reality, what we've done is we've gone from cuboid epithelium, a single layer of cuboid epithelium lining this duct to the inside in the proximal convoluted tubule, to a layer of simple squamous in the thin descending limb of Henle, continuing as simple squamous at the bend in the loop of Henle and the thin ascending limb of Henle. And then as we move upward, we're going to convert back to cuboid epithelium in the thick ascending limb. So as a reference point, remember that simple squamous is an excellent tissue for filtration and diffusion, and simple columnar and cuboid are tissues that are secretory and absorptive. And so what we're going to see is a function change from the thin parts to the thick parts. As we follow the thin ascending limb upward, it passes by the glomerulus and then begins to fold again. So now this is folding pattern we say is convoluted again. But now we're further away from our glomerulus where we started, and the Bowman's capsule. So this becomes the distal convoluted tubule. So we have a proximal convoluted tubule, thin descending limb of Henle, thin ascending limb of Henle, thick ascending limb of Henle, and distal convoluted tubule. Now, a distal convoluted tubule connects to a much bigger tube where other distal convoluted tubules are connecting, so it's called a collecting duct. And the collecting duct is collecting the filtrate that's becoming urine and then getting ready to pass it on. So the filtrate would be flowing downward through the collecting duct in the final stages of being processed, and then eventually end up in the area of the papilla that we talked about. And so the duct here, where several collecting ducts come together, is called a papillary duct. And then the papillary duct would drain urine to a minor calyx that would be under that. So if we look over here, here is the same pattern, a little harder to see. My Bowman's capsule, proximal convoluted tubule, descending limb of Henle, ascending limb of Henle, distal convoluted tubule, collecting duct, and then papillary duct. And you can see how the papillary duct would drain urine into this minor calyx. So we were just talking about nephrons, and we were looking at the tubes, proximal convoluted tubule, descending limb of Henle, which is thin, ascending limb of Henle, which goes from thin to thick, distal convoluted tubule, collecting duct, and a papillary duct. So all of those collectively with this round structure, the Bowman's capsule, makes up a nephron. Now the loop of Henle is a countercurrent multiplier, and it allows us to adjust water balance and either make our urine dilute or concentrated. And it allows us to trade urea as a waste product for things that we want to reabsorb, like sodium chloride and other ions. So the loop of Henle is critically important. So if we look at this loop of Henle, notice it's short and it doesn't go very far into the medulla. Notice this loop of Henle is long and it goes way down into the medulla. So the second thing to notice is the Bowman's capsule for this one that's short is toward the outer part of the cortex, and the Bowman's capsule for this really long one is right next to the medulla. So because of that, these two nephrons have different names. So we refer to this as a cortical nephron because the proximal convoluted tubule, part of the loop of Henle, the thick ascending limb of Henle, the distal convoluted tubule, are all in the cortex. Over here, most of the loop of Henle is in the medulla. This is the Bowman's capsule is sitting next to the medulla, so this is called a juxtamedullary nephron. So we have about 80% cortical nephrons and about 20% juxtamedullary nephrons in our kidneys. And these concentrate urine a lot higher than these. So we can't produce a high amount of concentrated urine. But if we compared ourselves to a desert-adapted rodent like the kangaroo rat, then we would see they would have a lot more juxtamedullary nephrons and fewer cortical nephrons, and they can significantly concentrate their urine. So another way you could see it is in a cat. Cats produce fairly concentrated urine as well. So what we're going to do next is we're going to look at this structure of the Bowman's capsule and what's inside of it. And then I want to point out something that we're also going to talk about as well. So notice that as the thick ascending limb passes upward, it and the distal convoluted tubule come very close to this structure, which is our Bowman's capsule. And where they do, they actually pass over a blood supply. And we can see that here, where there's this blood vessel that they're passing next to right here. And so what we call this apparatus where this tube and this blood vessel interact is called a juxtaglomerular apparatus. So juxtamedullary nephron next to the medulla, juxtaglomerular apparatus next to the glomerulus. So the juxta in both of those names is referring to next to. All right, so what we're going to do is we're going to look at this structure in detail.